So today we are going to continue with our transport and plants section. So last day we covered cellular respiration and photosynthesis. I'm just gonna add 3 more here. Good morning. Okay, so we're gonna get going here. Thank you guys for tuning in. So yesterday we talked about cellular respiration and photosynthesis. A lot of you guys nailed the last question there. What is the relationship between the cellular respiration reaction and the photosynthesis reaction? And remember, photosynthesis, the reactants of photosynthesis are carbon dioxide, energy, and water, and it will produce oxygen and sugar, okay, as organisms that cannot produce their own energy like us humans. We need to acquire our energy from the food we eat, okay? So the food that, or the sucrose, essentially, that is created by plants will fuel our bodies so we're able to locomote, interact with our environment, and eventually reproduce. That is the whole goal of biology, is just to reproduce and to pass down your genes. One particular question that the majority of the class struggled with was when we were showing the specialized cells of a plant. So for example, a lot of you guys wrote down root hairs, which is excellent. And in the question, I wanted you guys to focus on the structure-function relationship there. And so the structure of root hairs are very small and there's lots of them, right? And the function of that is to increase and maximize water absorption or nutrient transport at the level of the roots, okay? Another specialized cell that we're going to talk about today is the cuticle. Okay, so remember, the cuticle is a thin, waxy material that is, just gonna admit one more. So the thin, waxy cuticle, or the cuticle is thin and waxy. So it's thin to maximize the light transmission that is coming in from the sun. So the structure is a thin cuticle, and the function there is to maximize photosynthesis. Okay, so if you guys are still not really sure with the structure and function relationship of some things, that is something that we should look into a little bit more. Okay, that's a really important relationship, not only in Science 10, but as you get to higher and higher levels in sciences and biology in particular, if you are looking to go into a biology related field. Okay, so today's topic is leaf tissues and gas exchange. Okay, so let's get into it. We're going to actually talk about some more specialized cells that allow the plant to better survive in its environment. Okay, so gas exchange in leaves, this is important as photosynthesis requires carbon dioxide as a reactant, and it produces oxygen, right, which is air. So this air, or like the carbon dioxide and the oxygen that is required in photosynthesis, it needs to be able to like travel through the plant. Okay, it can enter by passive diffusion. So remember, passive diffusion, it does not require energy. It goes from high concentration to low concentration. Okay, so if plants just had air entering by passive diffusion, it would take a really long time. And this actually would reduce the amount of photosynthesis the plant would be able to complete. And this would harm the plant because it wouldn't be able to get as big as it needed. And the bigger the plant gets, the more sunlight it gets, so the bigger it can get, okay? Yeah, so a leaf has specialized cells. So these are the guard cells to maximize its ability to exchange gases and transport nutrients throughout the cell. So guard cells are really important there. Guard cells will form stoma or stomata is the plural term, stomata. And we are going to talk about how that actually functions today on a cellular level. Okay, so just a refresher here, we've talked about the 3 types of tissue. So remember, we have ground tissue, dermal tissue, and vascular tissue. Those are going to be the 3 main types of tissue for plant cells. The dermal tissue in particular, this is the epidermis on the top and the bottom of the leaf. This is clear and very thin. Again, to maximize light transmission, so to, again, maximize photosynthesis. The epidermis is present though to prevent that water loss within the leaf, okay? Guard cells will form tiny openings called stomata, and this is gonna be mostly on the underside of the leaf, okay? So here can be another structure-function relationship. So the structure of the leaf is that the guard cells are on the underside of the leaf, okay? Now, remember, hot things always want to rise, sorry. So in the event where water wants to transpire, okay, that is what it's called when water goes from leaves into the environment. It's called transpiration. So transpiration, things kind of like always want to rise if it's warmer, right? So think about this, if you have like a hot shower, right? All the steam is going to the top of your bathroom, right? So kind of the same idea here. So heat is going to want to rise. So if we had these stomata on the top sides of the leaves, they would have more transpiration occurring. So these guard cells are mostly found on the underside of the leaf just to prevent that water loss. Stomata, so this is influenced by guard cells. These will control the rate of gas exchange depending on environmental conditions. So if it's really hot and dry, right, these stomata may close just to prevent excess water loss. Okay, so this is a cellular level of what is actually occurring in the stomata. Okay, so I'm just gonna get this out of the way. So we're gonna start from the top up here, okay? So step 1, we have sunlight, or it could be artificial light, okay? We have light striking the leaf will stimulate the guard cells to gather potassium ions by active transport. So this is going to be active transport because the concentration gradient within the guard cell and outside of the guard cell, there's going to be more potassium outside of the guard cell. So that's why it needs to spend energy to gather the potassium ions into the actual guard cells themselves. Okay, so that's step 1. So light striking the leaf gets the guard cells to accumulate potassium by active transport. Step 2, so once there is more potassium inside of the guard cells, now we're going to have a concentration gradient on the inside and the outside of the guard cells. So water is going to enter, it'll enter by diffusion because it wants to minimize that concentration gradient. But remember, anytime water crosses a semi-permeable membrane, we consider that to be osmosis, okay? So water's entering by diffusion because there is a concentration gradient. But remember, anytime water crosses a semi-permeable membrane, we consider that to be osmosis, okay? But having that concentration gradient will force water into the guard cells, okay? Once water is in there, this part might be kind of hard to conceptualize, so I'm going to give you guys an example. So sometimes if you're out on a lake, you'll get those like inflatable donut tubes, right? When those are not filled with air, they're just kind of like, they fold over on themselves and there's no hole in the middle for you to go through, right? But once you put air into that tube, it kind of like swells up, and then will see that donut hole in the middle of that floating tube. This is kind of the same idea, where we have water entering the guard cells, and when the guard cells become filled with water, they just kind of swell up, just like that inflatable tube. Okay, so water follows the potassium, guard cells become turgid, and then the stoma opens. Okay, once the stoma is open, that is when we're going to have increase in gas exchange. Okay, after the sunlight is done activating or stimulating the guard cells in the leaf, the potassium cells, sorry, so that was step 3. So I'm just gonna jump, okay. Sorry, I got a little bit ahead. So we talked about this, right? So the water comes in, it swells up the guard cells, and then the stomata is going to open. Oh, we're not going to talk about when they close. Okay, so I'll just continue talking about that. So once the sunlight leaves, then the potassium ions are no longer being actively transported into the guard cells. So the potassium leaves, the water will follow the water follows the potassium. That turgid stoma will then kind of collapse in on itself, and then that will reduce the gas exchange. That stoma will be closed. Okay, make sure you guys are kind of familiar with this 4-step process. Okay, so maybe put a big star on this slide, just so you guys are knowledgeable about this concept. Okay, so this is kind of like a cellular process. If you go into Bio 20, you will learn more in detail how these cellular processes are influenced. Okay. Okay, so just kind of a summary of what we just talked about. So guard cells are necessary to prevent excess water loss. So this is known as transpiration in plants. When the water leaves the guard cells, the stomata closes, as we talked about. And plants that live in hot and dry environments will have less stomata compared to those that live in humid environments. Okay, so I want you guys to think about this for 10 seconds. Gonna clean up our attendance. Cool. Good attendance today, guys. Excellent. Okay, let's go back here. Okay, so plants that live in hot and dry environments will have less stomata, because if you have less stomata, it is just less opportunity for you to be losing water, right? It's like if you wanted to keep your window, or if you wanted to keep your house warmer in the winter, if you wanted to keep your house warmer in the winter, you would probably try to minimize the amount of openings towards the outside, right? Like you wouldn't want to have a bunch of windows that are open, right? You'd want to close all of your windows and possibly reduce the amount of windows that you have, just to keep the heat in and to keep the cold out. Okay. So ground tissue, I know you guys have talked a lot about ground tissue, and I think you guys have a really good idea of what ground tissue is. Now we're going to talk about some types of cells that are in the ground tissue that have particular functions. Okay, So first off, we're going to talk about the mesophyll. So that is the layers of cells between the dermal tissue, okay? So we have dermal tissue, and then just in general, we have the mesophyll, okay? Within the mesophyll, we have palisade tissue cells. And these are the sites of photosynthesis. So I remember that the palisade tissue cells are the sites of photosynthesis because they both start with P's, okay? So the palisade tissue layer is actually the kind of region really close to the top near the epidermis. And there are lots of chloroplasts found here. Of course, that makes sense because chloroplasts are the site of photosynthesis in plants, okay? And I also want you to focus on the organization of the palisade tissue layer, okay? They're really tightly compacted, okay? And again, this is to maximize photosynthesis. So if we wanted to do a structure function example, the structure of the palisade tissue layer at the top, like near the epidermis, is really really tightly compacted. And again, so that would be the structure, and then the function of that is to maximize photosynthesis. Okay. Yeah, so let's move on to the spongy mesophyll tissue. So of the mesophyll, you'll have your palisade layer at the top. So these are where the palisade tissues are. And then the spongy layer down here, we're going to see that these cells are kind of packed not as tightly as the palisade tissue layer. And this is because this is the type of tissue that is kind of setting up, it's making it easier for gas exchange and diffusion to occur throughout the leaf, okay? So these are going to be loosely packed. So the structure, they're loosely packed, and that is to maximize gas exchange and diffusion throughout the leaf. So there's another example of a structure function relationship. Okay, this thing right here is a vein. Okay, this is part of the vascular tissue, right? We have xylem and phloem in here. Remember, xylem transports water and phloem transports food flows, right? All right, so I want you guys to examine this. This is a image taken of a leaf in a microscope. So it's about 70 times magnification. So behind my computer, I'm looking at my avocado plant, and I'm just kind of envisioning this inside the leaf. So in reference to what we talked about last slide, where is the mesophyll area? Where is the spongy mesophyll layer? And where is the palisade tissue cells? And this is the top of the leaf up here. So top of leaf. So we, up here, so we have our epidermis, excuse me, our lower epidermis. We have stoma and guard cells here. So we're gonna have our palisade tissue cells nicely packed really tightly up here. Okay, so the mesophyll area is just the area between the cells, between the epidermis. So this is going to be our mesophyll area. Palisade tissue cells are going to be right near the leaf surface, right, to maximize photosynthesis. And the spongy mesophyll area is this kind of like holy, yeah, so it's kind of like the spaced out area underneath. And remember, this is to allow for maximal gas exchange throughout the leaf. Okay. So you should be able to identify these 3 areas if I ask you on an assessment. Okay? Cool. So we're going to talk about vascular tissue a little bit here. I'm just going to clean up my slides. Okay, so same thing. This is just not a microscopic view. This is like an animation of the leaf, a little bit easier for us to see the organelles. So vascular tissue in general, this is what's going to transport food and water throughout the plant. So again, xylem produces, or xylem tissue provides the leaf with water and minerals needed for photosynthesis. We're going to talk about water transport more in next lesson. Okay, and then we have the phloem, which will distribute sugars formed during photosynthesis. Xylem and phloem are usually bunched together, and we call these vascular bundles. I would encourage you guys, if you have celery at home and food coloring, you can put the celery into a cup of water. And if you put in a few drops of food coloring and leave it for a couple days, eventually you will see that colored water will be transported up these vascular bundles, and it will dye the vascular bundles of the celery. It's pretty cool, actually. And if you have any celery at home, I would encourage you to do that. It would be a really good way to just kind of visualize what we have been talking about today. Okay, so that is it for today's topic. So I'm just going to end the video here.